Microtubules are tubes around 25 nanometers in diameter. In these pictures, they're colored green. They form a highly dynamic star-shaped structure called an aster, organized around the centrosome. Microtubules anchor and move the chromosomes during mitosis. They also position the organelles within the cell using proteins known as molecular motors.
Der Unterschied von dieser Rekonstruktion zu der tomografischen ist, diese basiert auf Mittelung. Ja.
This electron microscope provides a high-resolution image of the microtubules. The walls of the microtubules are composed of repeated units of a protein called tubulin. Very thin sections taken from a yeast cell make it possible to visualize the way in which the microtubule network is organized.
And here there is one microtubule who goes the whole way to this tip. In this yeast cell, I think usually they say between 20 to 40 microtubules in a cell, and in a eukaryotic cell you would find much more microtubules, of course. When I have the microtubule oriented like this, I can model it. And so what I want to do is a 3D dimensional model of the microtubule cytoskeleton and see how the microtubules are organized in comparison to each other and everything else inside of the cell. You see how it created the line there? When I have all these lines put together, it becomes a model. And this model is now 3-dimensional. So you can see nicely the details of the cell, and on top you can see the models. For example, in red here, the nuclear envelope, and then you have the microtubules laying around it. I can vary this as much as I want, and then I'm doing, I can zoom in as well and look at specific details. For example, all the red ends is a capped microtubule end, which means it's a minus end. And all the blue balls, where I'm putting them, for example here, means it's a flared end, so it's probably a growing end of a microtubule. So what I do, I take these thin sections, I have a tomogram of each, and then I make a model of each, and then I join them all together. And so then I have a really large volume of the cell, and we can twist and turn as much as we want, and we can zoom in on details that we find interesting. So for example, if I take away the plasma membrane, it's easier to look at the microtubules. And then here you can see how the bundles of microtubules are laying parallel to each other. And here in pink, I have modeled the centrosome. And you can see how the microtubules are laying straight on top of it.
Or is it not? Yes.
I mean, now you have one cell anyway.
There are other ways of investigating a living cell. Using lasers and fluorescent molecules, it's now possible to monitor the way molecules behave and interact within the very heart of a cell. Même d'une cellule.
On regarde dans une dimension, disons, d'à peu près un millionième de millimètre, disons là où la vie des molécules se passe. Et pour bien observer ces phénomènes, au fait, on doit aussi reculer dans le temps. Il faut carrément regarder dans des temps extrêmement courts. Donc le microscope que vous voyez ici est au fait désigné à observer des phénomènes sur des nanosecondes. But the cell is basically a shape in space. And the question is, how is spatial regulation of dynamic structures organized? So we want to know if the cell is capable of generating localized environment or environments that will actually direct the microtubule cytoskeleton in an asymmetric way, thereby actually directing shape. There's a local environment where this molecule is inactivated and therefore promotes microtubule growth. In other words, where the cell will move, the microtubules will grow into the extending lamellopodia. So in other words, a local environment is created at the leading edge of the cell that will actually direct the microtubules towards where the cell is moving. So now we have localized activity, not at the membrane, but on the chromosomes. A very similar principle applies to mitosis, where now what we need to do is actually direct microtubules towards the chromosomes in order to pull them apart. So again, we have a situation of asymmetric growth. Now this is a typical mitotic cell in metaphase, where we have a nice asymmetric growth towards the chromosomes. But again, if we look now at the state of this molecule that regulates microtubule growth, we can see here that again it's in this floppy conformation, not interacting with tubulin exactly around these chromosomal structures. So basically a gradient around the chromosomes generates a steady state structure that will direct the microtubules towards the chromosomes. So the cell is now capable to generate a local environment by localizing activities on these macromolecular structures like chromosomes.
How are molecular motors attached to microtubules, and how do they move? These motor proteins, called kinesins or dyneins, transform the cell's chemical energy into mechanical energy, keeping things moving within them, and providing a new line of investigation for researchers. Dresden, Max Planck Institute. Explore.
For nanotechnology, we have turned the concept of the motor and the microtubules on its head. And instead of having the motor walk along the microtubules, what we're doing is we're having the motor move the microtubules. In this case, what we're doing is, or what you see here is the microtubule moving. It's moving in a trough that's about 1/1000th of a millimeter wide. And because it's hitting the walls, it doesn't move outside of this area. We hope to be able to manipulate the direction and the locations of the motors such that we can manipulate small molecules in the nanometer range.
And some biologists are already dreaming of micro-machines driven by molecular motors.